name is Mike Esposito. I'm a microbiologist over at MCR Labs. Prior to my time at MCR Labs, I worked as a cultivator in a few different operations within the state of Massachusetts. As a cultivator, I became very familiar with microbial contaminants within the grow. For those of us who've dealt with microbial contaminants within the grow, we understand how stressful of a situation this can be. Fortunately, there's a relatively cost-effective way where you can go about determining exactly what species of organisms are causing your samples to fail. This information can help you understand how this organism got into your grow, and additionally what steps you can take to remove or remediate it from your grow. This process is known as speciation. It all starts back in 1977 with the advent of Sanger sequencing. This technology allowed researchers to determine the actual genetic sequence of amplified DNA. In 2003, researchers at the University of Guelph first coined the phrase molecular barcoding. Molecular barcoding is the practice by which common shared DNA across multiple species is sequenced via Sanger sequencing, each generating its own unique molecular barcode. Let's take a look at where this technology is used in real life. In practice, large databases like the Basic Local Alignment Search Tool, or BLAST database, are utilized to reference sequences that have been acquired by scientists all across the planet. These acquired sequences can then be compared to a new organism where a molecular barcode is obtained. This technology is very useful in determining things like individual species within a cryptic species complex. In the case of a cryptic species complex, we may see multiple species that look identical to each other. In cannabis, this is often found with things like white filamentous fungi. Through the use of molecular barcoding, we can determine which member of that cryptic species complex you're actually looking at. Because these different species have very different properties and traits, knowing the exact member of your cryptic species complex can guide your path forward towards remediation and removing this pathogen from your grow. Here at MCR Labs, we use speciation to help our clients to determine exactly what organisms are present in their failing or near failing samples. This is really useful information to clients as it can help them guide their IPM or integrated pest management approaches. Every microorganism is different and they all have their unique Achilles heels, which a grower can exploit to remove or remediate the issue from their grow. Now that we have a better understanding of what speciation is, let's take a look at this service in practice. Let's say that you've submitted a sample for testing. A few days have passed and you've received your results. Unfortunately, your sample has failed for fungal contaminants. At this point, you could reach out to our client services department and request our speciation offering. Once speciation is requested on a sample, we'll begin by culturing out any organisms that are present on your material. This means we will apply your sample to a grow media that's very amenable to fungal reproduction. Once there's enough reproduction and growth of new fungal organisms, we will subsample from there, and that's where the genetic portion of speciation begins. This portion starts with lysis. Or cracking open the cell walls of the organisms that we're trying to identify. Once lysis is carried out and DNA has been extracted and purified, we can begin by using PCR, or polymerase chain reaction, to amplify millions of copies of the target DNA. Once we've obtained these copies, we can send them out to a third-party lab for sequencing. This lab will sequence and return results to us in a sequence file. Using that sequence file, we can compare the molecular barcode generated from your target organism with a large database like the BLAST database, which contains millions of accessions, or previously sequenced samples, from all across the globe. Using this reference library, we'll be able to determine exactly what is present in your sample and allow you to move forward from there. Once you receive your report, you'll have an idea of exactly what species was present on your sample, as well as a brief description of the species and some approaches you might take to remediate it out of your grow. If you have any further questions, you can reach out and a member of our microbiology team will be happy to discuss the specific organism with you and what steps you might take to remove it from your cannabis cultivation. Through the use of speciation, we can help not only individual clients, but the larger agricultural community as a whole As we see cannabis material increasingly transported from state to state, as well as changes in the climate and ecosystems around us, it's important that we're able to monitor and determine what organisms are present on cannabis material in certain regions. The use of speciation can allow you to sort of keep a watchful eye and get an early idea as to what organisms are being introduced to a new region. With this information in mind, you can plan around this before these new invasive organisms have a dramatic impact on the agricultural range around you. If you've taken anything from this video, here are 3 main points you should consider. First, speciation is the use of molecular barcodes, or very specific genetic sequences within all organisms, that can be referenced against a large database collected by scientists all throughout the planet to positively identify exactly what species is present on your sample. 2. The use of speciation can be wildly beneficial to a cultivator, as knowing the exact organism that you're trying to combat can allow you to strategize and tailor your approach to remove this problem from your grow so that you don't have to deal with it in the future. And 3. The use of speciation is not only beneficial to the individual client or cultivator, but to the larger agricultural community as a whole. Having an advanced look at when invasive species make their way into a new range can allow agriculturalists to plan ahead before these new invasive species devastate the ecosystem around them. If you have any additional questions, please feel free to reach out to hello@mcrlabs.com. Additionally, links to the website are available below.